bit long. I will talk for an hour, and as you can see, I will talk about the microbial web and its plankton and processes and how does this go? Okay. I will start with an overview on microbial food web and the plankton dynamics. Then I will explain the challenges proposed by the dynamics of marine microbes. And I will very briefly talk about the main elements of an ecosystem model, because as you already did a very good job yesterday. But I will focus a little bit on the drawbacks of modeling, but I will also talk about the solutions and how we should proceed in the future. And I will link the marine microbial food web modeling to zooplankton a little bit. I thought this was essential if we are talking about integrated Earth system models. Really, zooplankton is the link between the lower trophic and the higher trophic levels, and especially if we are going to add the human dimension on top, this is really a must. Okay. This little loop that you see on the left is the main element of the food web dynamics in the oceans. And autotrophic prokaryotes, small phytoplankton, can directly feed zooplankton, or they can indirectly feed zooplankton through coagulation. For example, zooplankton can go eat detritus that's made of phytoplankton. And I will go into more detail about this is a very important part of the food web dynamics in the marine system, and this is the main driving force of the nutrient dynamics in the ocean. Okay, as I said, the pelagic food web is microbe-centric. When we talk about food web dynamics, we generally talk about the diatom copepod fish, but this is really a small part of the marine food web. And the microbial food web includes the small microzooplankton, unicellular zooplankton as well. And if we add up the biomass of this microbial food web, it's 5 to 10 times the mass of all multicellular marine organisms. And if we go ahead and if we look at the metabolism, the energy throughput of these cells is very, very high. If we add up enough cells that would equal the biomass of 100 humans, the energy throughput would be equal to a nuclear power plant. So the energy that goes through at the microbial level is very, very high. Okay. Maybe you've seen this diagram before. The frontier of microbial food web dynamics was Kazan, and this was an illustration from this paper. And as you can see that really the microbial food web is composed of that plus living cells. And the living parts are called the hotspots. For example, bacteria or yeast. The smallest ones are viruses, and it goes up to ciliates, which are up to 2 millimeters in size. Okay, this is an example of a real food web. This is a marine food web. As you can see, it's extremely complicated. But in order to model this, we need to simplify it. There are some examples here. This is an illustration of Nemo Medusa, and this is the ERSA model. So what do? We end up aggregating similar compartments in the food web into compartments in models. For example, when we talk about zooplankton, we generally talk about small and large zooplankton in models, and just a few types of phytoplankton. However, in nature, there are hundreds and thousands of types of different species. So we just lump them together. And at the end, this introduces some errors, but this is expected. We need to somehow simplify nature if we are going to model it. Also, in terms of community structure, I forgot to say, it's hard in models to capture the changes in the community because we are missing many elements of the community in the models. I will step back and I will give you a brief overview of what we are modeling when we are doing microbial food web model. Okay. This is the phytoplankton cell and this is the bacteria cell. Okay. How do they work? These are also models of these cells, right? This is the simplified version. Phytoplankton uses energy from the sun and it uses inorganic nutrients to synthesize carbon, and in return it gives back, it produces oxygen. This is the main photosynthesis dynamics. On the other hand, bacteria can use dissolved organic compounds, and it uses oxygen and gives out carbon dioxide, and it also produces, it can produce inorganic nutrients. How does the synthesis of carbon work? This is a simple illustration. Think about two elements. This can be nitrate or phosphate, A and B. You need enzymes to capture these two, and at the end to synthesize C. Okay? So the limiting nutrient defines how much C is synthesized at the end. In the cell, the enzyme waits for both to be available, and then it synthesizes C, and if A is missing, it continues waiting. Okay? Again, this is also a simplified version of what's happening in the cell, but in terms of, but we need to somehow mathematically represent this, and we use the Michaelis-Menten dynamics to do that. Here you see how production rate changes as a function of substrate concentration. This can be nitrate or phosphate. We estimate the house saturation concentration in laboratory or in situ experiments, and if we know the maximum uptake rate, we can put this into mathematical formulations. Okay. The same thing goes for light. The cell captures photons with its chloroplast, and this is quite a complicated process. But again, we simplify it. In high light concentrations, the photosynthesis goes to equilibrium. At the beginning, it's quite steep. Again, this is a Michaelis-Menten type curve. And at low concentrations, the cells tend to increase their chlorophyll concentrations. So at low light, chlorophyll to carbon ratios are higher compared to high light regimes. Okay. So after setting up these equations, we need to include this in ecosystem models, in a full model. In most models, in most MPZP models, the population growth was estimated as a function of in-situ nutrient concentrations. Here, you can estimate the biomass in a meter cube of water, for example, if you know the nutrient concentrations in the cells, and if you know how many cells exist in that water. And you can have a simplistic equation like that to estimate the biomass in a cell. This can be a function of maximum uptake rate and the in-situ nutrient concentrations. This is the Michaelis-Menten equation, and then you need to, of course, extract how much you lose through mortality. And alternatively, you can estimate how many cells you have. And this is an equilibrium between growth and mortality. And growth is, can be estimated as a function of maximum growth rate, and this can be a function of temperature and gradients, but it should be a function of what's in the cells. Okay, the growth in nature is based on what's in the cells, not what's in the water. But when you go to an ecosystem model, like if you are going to have different functional groups, different types of zooplankton, you need to combine these two approaches. Okay, at least that's what I did. I estimated uptake of nutrients as a function of in situ nutrient concentrations, but also intracellular nutrients in the cells. And then the growth was estimated based on what's in the cells. Okay, depending on your system, you can pick which equations that you can use. For example, the first equation that I have shown you, the biomass as a function of in situ nutrients, can be used if the nutrient concentrations are very low and if the system is in equilibrium. And at the end, you can estimate the nutrients, what's left, because these organisms remove as much as nutrients as they can until no one else can use it. Dinoflagellates and cyanobacteria are well known about this. These are smaller cells, and then the nutrient concentration can be estimated using the Hall saturation constant and the uptake rate and the intracellular nutrient concentration. These can be called cleaners. They scrape off what's there, and then at low nutrient concentrations they may have an advantage. But if we are talking about diatoms, for example, they grow in high nutrient concentrations, they can take advantage of that. And then this time you need to know how much nutrients they can take in their cells, because they take up excess nutrients, even more than they need. This is called the luxury consumption, and they can use it at a later stage. And so an organism can move, can increase its growth through increasing its nutrient uptake, or it can develop, or if it has a better defense mechanism, then its number or its biomass can also increase. Okay? And as I said to you, as I told you, if we are going to capture the different types of phytoplankton under changing environmental conditions, it's better to combine these two dynamics. I will go over the main elements of an ecosystem model, as mentioned yesterday, but I will go into more detail here. If you are going to set up an ecosystem model, you need the governing equations, okay? And generally these are mass conserving. You need some initial boundary conditions, and external forcing. An example is a model that I have developed for Sargasso Sea. Here we had, actually this is based on another model that I developed for the Equatorial Pacific when I was a student in IB. And in this one we use the Drude equations, and as I mentioned to you, these equations use both intracellular concentrations as well as ambient nutrient concentrations to capture uptake dynamics. And there are two types of zooplankton, so all the zooplankton are lumped into these, and different nutrients and dissolved organic matter, and in practice it's quite simplistic. But in order to make this model run, you need to have a governing equation. Generally, the governing equations are composed of the physical part on the left here. You need to have advection, diffusion, then